Thank you. So again, I want to thank everyone, all the organizers, Forrest Donka, everyone, and all the speakers. This has been a great event. Particularly the art and music last night was fantastic. I really enjoyed that. So I want to give you, I only hesitantly raised my hand last night when they said, if you're a phage biologist. So I am a soil microbiologist. I mainly study bacteria. And yes, phage food, right? So, and just a couple months ago, I was starting to play with, I was on a plane ride, and I thought, oh, I have this old, couple-year-old viral metagenome that I haven't looked at because it was hard, right? I'd looked at the bacterial, the bulk DNA metagenome from these soils in the Arctic, and I started playing with it just in time for me to be able to submit an abstract to this symposium. And so I'm going to basically give you the caveman perspective. This is what happens when someone like me stumbles into viral metagenomics and just has to try to figure out from this sort of dark forest of, unidentified genes, what's going on in my ecosystem. So you can even tell I probably put the wrong number of tail fibers on that thing. Okay, so this started a little history. When I first arrived at SDSU, I was teaching a microbial ecology class. The first time I've taught about 10 times now. Forrest was a guest speaker. And I made some statement, it's sort of innocuous. I was probably introducing Forrest and said something like, phages are important predators in the ocean and may be important in soils. And of course, Forrest is like, maybe important? and again and again, I think his entire lab for, I think years, I think Maya continued to tease me about saying maybe important. So, but eventually, some people from my lab and Forrest's lab did publish this paper on top-down control in our system in the Arctic. And it's soil, but it's practically a wetland. These are really organic, rich soils, very, very wet. And phage are certainly important in regulating bacteria in this system. And so, I'm convinced that they're important. So eventually, I'd like to know who they are. And so with the help of Liz Dinsdale and her students, and again, this is only my rough reconstruction of what's happened, what they did. I need to go through my technician's lab book and reconstruct it. But this is what I think they did. We had soils, the same ones I'd used to make the soil microbial metagenome from the bulk DNA that was extracted from these soils. Presumably, we extracted the viral fraction, concentrated them at density gradient, DNA, all the free DNA, and there was only a small amount of DNA at that point, so it was amplified a little bit. Shotgun sequencing on the old 454 platform. And then, like I said, we also have, from most of these same soils, microbial metagenomes made from the same soils. So we consulted the great and powerful mgRAST. This is the software that's, that's Oz. That's mgRAST. Rob's not here, so I'm making extra fun of him. So I love, this is a server, many of you have probably used this, so great for annotating metagenomes, and normally we have things uploaded there and it just tells us all the answers and then you're done. But obviously, in this case, the great and powerful mgRAST said, reply hazy, try again later. It's, as you've already heard, there's problems annotating viral genomes. And so this is one example, one of our metagenomes. Started off with, so there's a lot of these failed the quality control initially, but still we started off with 66,000 good sequences to start with. And of those, most of them produced predicted protein features, 21,000 odd different protein features. And of those, only 1,340 were annotated. And that represented about 4,500 sequences. And then even beyond this, so we're already down from 66,000 to about 4,500. And half of these or so were even classified by mgRAST as things other than viruses. So if I was just going to say, okay, mgRAST, tell me about the viruses, now I'm suddenly looking at 2,000 out of my 66,000 sequences. And again, being a caveman, this is a problem for me. So this gives you some of the other libraries. The idea, we have a depth profile going down The permafrost in this environment is around 30, 35 centimeters deep, so that 30 to 40 represents a transitional zone. Some of that, the bottom parts of it, some or all of it has not been thawed for, I don't know, 10,000 years, something like that. So you can see in general how poorly, so we start off with this many sequences, and if I'm just looking at the things that NGRAS tells me are viruses, it's a pretty paltry number. But looking at these viruses, the first thing that popped out at me was, well, they were mostly bird viruses, which is very interesting. So sorry about that, phage people. I'm going to be talking a lot about bird viruses too. But algae, plant, animal, and certainly bacterial viruses, phages. So at least let me start off by talking about skimming the cream off of this data set, the stuff I could get out of NGRAS very simply. Oh, by the way, of those unclassified viruses, I think a lot of these I can actually classify, and a lot of them are circovirus-like. Probably, they look a lot like Maya's circovirus-like genome. It's fun looking on databases because you get to see what your old friends are up to. They put up sequences, you're like, yeah, that's Maya's sequence. NE, does that sound familiar? A genome named, anyway, it was one of yours. A lot of them showed up in the Arctic soil, so Florida, I don't know, Arctic.
I travel there all
the time. Yeah. It's not your fault. Here's just, so again, I also found viruses in the microbial metagenomes, that is the ones that were just made from taking the bulk DNA of the soils. And those metagenomes I've already published on. They're far better quality, really easier to annotate. But you can see basically, in this viral fraction floating around, presumably viral-like particles, you can see it's dominated again by the circoviridae, which are mostly bird viruses, and then some other animal viruses. And those are, so anyway, they're certainly within the microbial metagenome. It's dominated by phage. But when we look at this sort of viral particle fraction, a lot more other types of viruses. And here are just the list of the phage sequences in the viral metagenome that could be identified. Interesting about this list, chlamydia, these are not the most abundant bacteria in this ecosystem. The most abundant bacteria in this ecosystem are generally in the Nacido phylum. These things are very important in soils. They're very rarely cultured. They grow very slowly, and I'm actually starting to culture these things. The main thing is just to be patient. Just tape up your plate so they don't dry out, give very dilute media, and they'll grow up in about 3 months. Seriously. Literally about 3 months, 2 months. And so probably not the most fun thing to do genetics on, but we can at least sequence their genomes and look for phage in those sometime. That would be fun. I do want to point out that this Acinetobacter phage, that's kind of interesting. And Clostridium, there's certainly a lot of Clostridia, particularly in the lower layers. It gets really anoxic. It's a very wet, stagnant ecosystem. And so anyway, that's just description of what I found. So what was interesting is my process, and this has just started, so again, like I said, I'm essentially a caveman. I'm new at this, but I'm very determined like a caveman. So I'm going to basically crack this coconut with rocks or my head or whatever is necessary. So trying to make some headway into all these unknown sequences. So predicted proteins, downloaded the predicted proteins, and started doing local blast Ps, so protein blasts. And so this is sort of a reiterative approach. Found genes and genomes that were identified by NGRAS, and then just started looking for similar things or things from the same genome, other genes in the same genome. And that already has produced a lot of results. So taking, again, the most abundant ones were bird viruses, but this could apply to anything that was abundant in the data set. Beacon feather virus. Originally, there were 82 sequences. This is just for my worst metagenome, by the way, the summer one that had 300 annotated sequences, starting with some 30,000. So you can see that, and these weren't even, a lot of these had really nice, so this E value, these E values are in the same range of what MGRAST was originally annotating as well. So just using the same criteria that MGRAST had, I found E values. I was able to find, let's say, another 376 new sequences So increase that from 82 to 376. For canary pox, from 68 up to 260, et cetera. Circoviruses, found a bunch of new sequences. Again, that all had decent, some of these had better or certainly not worse fits to what was in the database already. These poor fellows. Very sad. So then I also want to look at these things that MGRAS classified as non-viral. And so they were mostly bacteria. So the question is, could they be contaminant DNA? That would be the first thing you'd guess. Or are they just incorrectly annotated viral genes? And at a glance, it's clear that most of these things are viral genes. Some of them are even prophage, a lot of rep proteins, proteases, things that are very conspicuous, I think, for viral replication. And also because I happen to have data on the abundance of bacteria from these same cores, I could see if there's any correlation. So if it's contaminant DNA, one would expect that the most abundant microbes would be leaking the most DNA. And so there was essentially no correlation. This is one example from the top 10 centimeters. Everything, so this is the rank abundance of the microbe core versus the viral core for the entire thing, and then just for the top 20, and you can see really no impressive correlation. Same thing for the next layer down. So I feel that most of these things are probably genuinely viral and aren't just contaminants. So let's look at them in a little more detail. Again, bacteria were the most common, but it was a really nice balance, a lot more diversity than I see in the microbial cores. So again, there's a lot of bacteria, but there's protozoa, algae, insects, archaea, plants, animals. So is it possible that these represent the hosts? That would be really convenient if it does. So Giardia was the most abundant one, and Cryptosporidia. There's even this microbial animal, Trichoplex. You can tell by the way that I personally love the biology. If I was a Drosophila geneticist, I'd like the Drosophila, and then I would get thrown off the mailing list within days. But the most abundant, in fact, all of the Giardia sequences were rep proteins. And then this Acinetobacteria, which again showed up in the actual phage annotations, it was the most common bacterium. And it was mostly rep A, replicase, so various kinds of nuclease hits. So again, to me, this suggests that these are actually viral genes that just were annotated as bacteria. So putting it all together, what does this tell us about the ecosystem? And that, by the way, is a little bird sitting on a frost mound. The permafrost does interesting things and shifts to the topography. So the thing that really struck me looking at this data, because again, this is a sort of new, I have not been doing this for long. I've been studying soil microbial communities. The viral DNA represented the entire ecosystem. So not just the soil microorganisms, but plants, animals, insects, protozoa, algae, and fungi. And so bird viruses dominated numerically, but they were very low in diversity. There were just, again, about 3 genomes that got hit over and over and over again. The phage genomes were by far the most diverse. So looking at the ecosystem, I've been up there for years, and the birds up there are magnificent. People, like the main tourism you see up in Barrow, Alaska, are people with these cameras that are this long, and they'll sit there crouched in the tundra in the wetness for all day. There's these beautiful snowy owls that perch up on the occasional power line. These Jaegers that are extremely territorial and will just scream at you when you're trying to do field work. Arctic swan, etc. So excuse me. So it's hard to ignore the birds, even if you're a soil biologist, especially since there's a lot of this. Beautiful bird poo all over the landscape. Notice this one here, the contrast of brown on green, the sphagnum, but then the reverse color scheme here with the green on the darker colored sphagnum. So if I'm sort of tempted, if I can get this into a coherent manuscript, to submit this as a cover photo for the journal, just like, because that's really the viriome of the soil. It comes down to that. But lots of other wildlife as well. So these beautiful, adorable little lemmings. And then this is what the lemmings look like after they've been eaten by an owl and spat out. That's an owl pellet. Other small mammals like that little ermine weasel. Larger mammals like caribou. And of course, occasionally the polar bears do make it, not just from the ice, but to the tundra. This one's hanging around one of the old dumps. But that's all surrounding tundra. And in mosquitoes. So, looking to the future. So first of all, like this theme has come up again, and I could tell from looking at posters last night, and I was hoping to hear Anka's talk before, well, hopefully Anka will give you a lot of answers about how to address these questions of the phage dark matter. I can say this reiterative approach has helped. Start with, so it's kind of like looking for the key below the lamp or whatever they call it, except for maybe we're looking just in the dark area, just outside where the lamp is. But basically, look for similar things to what you already know is there, and you start moving it outward. I've already at least, quadrupled the number of hits I could find. And of course, we need to find more about viruses for these, just like Lisa was just saying, the uncultured bacteria obviously are going to harbor uncultured phage. So earlier today, Forrest came up and said, hey, dirt boy, we need crazy stuff about the future. And so I had to add this next slide. And again, this is more about viruses than phage, but I was so impressed by how the soil virus fraction represented the diversity of the entire ecosystem. That occurred to me you could reconstruct the ecosystem level diversity maybe just from soil viruses. In Barrow, I've been to these ice cellars where you can go down. You don't need to have a freezer if you live in Barrow. You carve out some permafrost and you can keep your whale meat and stuff down there. And I've gone down to some of these and there's buried peat layers, 10,000 years old. You can drill in and probably find some preserved peat soils from the past and look for, stuff. Like I said, I'm a caveman, so this is naturally. Now, of course, the challenge is to figure out the bioinformatics problem before all the permafrost melts. So the clock is ticking. All right, thank you.